Hey guys, so this lecture is going to be on ligands and receptors in drug delivery. So I'm going to first talk about ligands and what they activate. So we want to define something as the effect. The effect is the defined outcome that is desired as a result of activation. Now, not all agents or ligands that interact with a receptor will trigger the sequence of events we desire. It's important that we define this effect as something that we desire because an interaction could lead to a result that is not desired. And so when it is desired, it's called an agonist, where the ligand or bioactive agent triggers the sequence of events leading to the desired effect with their interaction. Whereas if the desired effect does not occur with the interaction, it's an antagonist. So say I have a hypothetical agonist, and where the dose of the agonist affects what percentage of the max effect is. And so with my hypothetical agonist, I can expect a 100% of the max effect at a dose of 1 on this logarithmic scale. Now, depending on what type of antagonist I add to the system, the dose of the agonist will change to get the same percent of the max effect. And so the first type is a competitive antagonist, which I'll put in green, where even if I have a competitive antagonist in the mix, the concentration of the agonist can be adjusted to overcome the effect of the antagonist. And so what this looks like is that you'll see a translational change towards the right, where I can still get 100% of the max effect, but I would need to increase the dose of the agonist to get to 100%. And then we can have a non-competitive antagonist. However, with the non-competitive antagonist, the agonist concentration does not overcome the antagonist. And so usually what will happen with this is that you will never see 100% of the max effect, because the agonist concentration still is not enough to overcome the effect of the antagonist. And so you might see something like this, where it doesn't matter how much agonist you put, it still is not enough to overcome the presence of a non-competitive antagonist. The third type is kind of different. It's an irreversible antagonist, where the mechanism of action is just being changed, and this can be degradation, and so it could be phosphorylated into something that can't work anymore, or it could lead to protein breakdown. However, with irreversible antagonism, it's kind of similar to a non-competitive antagonist, because adding more agonist will have no effect to overcome the effects of an irreversible antagonist. And so when you do have a ligand or a bioactive agent that is an agonist and it binds to a receptor at the site of action, what is sometimes the desired effect is a signaling cascade, where the activation of the site of action triggers a result in A, which also triggers B, which also triggers C, which then leads to what we defined as the effect to be activated. However, if you wanted to block the effect without targeting steps A through C, how could this be done? And so a way that this could be done is using soluble receptors. So you can bind some dummy receptors to the ligand. So if you have a little receptor here, which I guess looks like little antibodies, if you have a bunch of dummy receptors, then the ligands would in turn bind to the dummy receptors that are soluble, rather than binding to the place that would trigger the site of action. And so these are just free-floating receptors that aren't on the membrane. And so please note that because the site of action is on the membrane, this method only works for membrane-bound receptors acting as the site of action. And so an example of how this works in the real world is in corneas. And so I have this picture of a manatee on the right because in most mammals and animals, corneas are not vascular. However, VEGF, which is vascular endothelial growth factor, VEGF, promotes vascularization, and it is present in the cornea during eye formation. And so animals overcome the effects of VEGF during formation by the cells in the eye releasing soluble forms of SFLT1. And this prevents VEGF from vascularizing the cornea because VEGF will then bind to this instead of the actual site of action, whose receptor is FLT1. S stands for soluble. And so in the case of manatees don't make this soluble SFLT1, and so VEGF is able to bind to their site of action on the cells, and so they get vascularization on their eye, and the corneas are vascularized, and then that's the reason why manatees have really terrible eyesight. So I'd like to start talking about receptor families. However, before I give you the information, I'd like to present to you this thought problem. So in the context of muscle contraction is very dependent on the amount of calcium that is available in the muscle to perform that contraction. And so the intracellular calcium concentration can be increased by adding exogenous calcium, or calcium that is from the outside of the cell, or from being released from intracellular stores like the sarcoplasmic reticulum. And so I want you to think about how we can increase the calcium concentration inside of the cell while I'm going through these receptor families that I'm going to cover over the next several slides. And so the first form of a receptor that I want to present to you is a ligand-gated ion channel. And so the binding of the ligand will trigger ion transport across the membrane. And so in our context of muscle contraction, we want this to be calcium. And so an example of this is the NMDA receptor, and this stands for N-methyl D-aspartate receptor. Now it is a glutamate receptor. It's a ion channel protein in nerve cells, and there are two subunits. And so if this is one subunit and then a second subunit, this is a protein that is on the cell membrane of nerve cells. And so if this is the intracellular space, and then this is going to be the extracellular space. The only way that this ion channel will open is if glutamate and glycine are bound to these subunits. Only then will the channel open to allow calcium to enter into the cell. And so this is in the case of nerve cells. So another kind of receptor family that I'd like to introduce to you is the G protein-coupled receptor. This is a very large family of receptors. They are membrane receptors, and they have an extracellular transmembrane and also intracellular components. And I'm sorry for that typo. And so G protein-coupled receptors have three main components. And so if you can imagine that this is a membrane, there is the receptor component that has the initial interaction with the ligand, and it has seven transmembrane parts, where if this is the receptor portion, then this part out here will interact with the ligand. And so this is the receptor portion of the G protein coupled receptor. And so the G protein part actually activates the effector protein. And so this is attached to this receptor protein. And so this is our G protein. It is usually a complex of proteins, so maybe there's like 2 or 3 or maybe even 7 parts to it, where it affects and activates the effector protein. And so the effector protein then comes onto the activated G protein to carry out the effector functions. And this can be a signaling cascade. It can be activated as a kinase or a phosphorylator. But this is the G protein coupled receptor with 3 components to it. So another type of receptor are transcription factors. Transcription factors are a little bit different in that they're often cytosolic, and upon binding and activation, they'll enter the nucleus to interact with nucleic acids and alter the rate of transcription. And so if you imagine that this is a cell, and you have little soluble factors in the cytosol. Here's the nucleus. Upon binding and activation of a receptor, which I guess always looks like an antibody, it'll enter into the nucleus, and then if this is my very ugly piece of DNA, it will actually bind to a promoter or an effector region of DNA and directly influence the rate of transcription of a gene. And so this gene can be, say, a protein like a voltage-gated ion channel. And so in this way, it can affect and facilitate more calcium transport into the cell if we're thinking about our context of getting more calcium into a cell for muscle contraction. So the last type of receptor that I would like to go through are receptor protein kinases. And so these are membrane-bound enzymes with a binding domain as well as an effector domain. Now I want to go through these terms so that you'll get a better understanding before I go through the context of muscle calcium concentration. And so kinases are proteins that phosphorylate other proteins. And then the binding domain of these enzymes are usually extracellular. And then the effector domain is intracellular, or at least intramembrane. And so in the context of our muscle thought problem, how can we increase the calcium intracellular concentration is by releasing the calcium that's stored in the sarcoplasmic reticulum. And this can be triggered by IP3. IP3. Now, IP3 can be produced by the cleavage of PIP2. And how this happens is that this is catalyzed by phospholipid C. So I'll just call that PLC. And the cleavage of PIP2 by phospholipid C becomes IP3 and DAG. And so what happens is that the receptor protein kinase gets bound by a ligand. There's a conformational change that occurs, and then it phosphorylates and activates PLC to increase the catalysis of PIP2 into IP3, and then increasing the release of calcium from intracellular stores, and therefore increasing the intracellular concentration of calcium. And so going back to our thought problem of how we can increase the calcium concentrations inside of a cell, we went through increasing calcium by opening ion channels from the outside. So you can use a ligand-gated calcium channel, as well as a G protein coupled receptor. If the effector protein is an ion channel or a kinase, or even it activates those intracellular storage releases. And then we can also have a receptor protein kinase that gets bound by a ligand, creating conformational change, and then phosphorylating and activating phospholipase C to increase the catalysis of PIP2 into IP3 and increasing the release of calcium from sarcoplasmic reticulum storage, and then thereby increasing intracellular calcium in the muscle to drive and promote muscle contraction. And so the next part that I'd like to talk about are second messengers. Now, second messengers are components of the mechanism of action that are downstream from the site of action. So you get the site of action, then you get second messengers, and then you get the effect. And so it takes the initial signal down towards the desired effect. And so there are two very large ways that this can happen. The first way with integration is that you could have different receptors. So if this is a membrane, you have different receptors, and these could be G-coupled protein receptors, it can be ligand-gated, but different receptors are activating the same G-coupled protein receptor. And this is just one example. And so the same cascade is activated first, and then, so this is an example of signal integration, where multiple different receptors being activated are all integrated into one single signal. And so another type is diversification, where, say, if you have a G-coupled protein receptor and it activates different effector proteins, multiple cascades can then be activated. And so this is a diversifying signal, whereas the integration version is where multiple different signals are integrating into one. In diversification, you have one signal that is diversifying into multiple cascades. And so a question that I want to ask is whether when you have second messengers in your mechanism of action, should you target upstream or downstream? Now, due to second messengers, you need to consider the adverse effects due to integration or diversification. So if integration is involved, multiple methods may be needed to block multiple pathways that converge into the same effect. However, if diversification is involved, if you block an upstream location, then it might lead to deleterious effects from blocking these multiple diverging pathways. And so an example is protein kinase C, where PKC is a second messenger of a lot of processes in the body. So if you block PKC, you may also block the pathway you intend to, but you also block the pathways that are essential to survival. And so you also see this a lot in developmental biology experiments, where if you're trying to knock out a certain gene, you might actually find out that that gene is crucial for a lot of different processes that ensure survival of the fetus. So there are implications of having second messengers on the dose-response relationship. And so if we assume that agonist receptor binding is reversible and proportional to the observed effect, we get three main equations. So in this first equation, the binding of a ligand to a receptor will lead to an effect, where the effect is kind of defined as LR for now, where this is the bound ligand to receptor leading to effect. The second equation is that the observed effect is proportional to the max effect, and this proportion is dependent on the ligand concentration as well as the affinity of the ligand to the receptor. And this affinity is also how easily the ligand falls off the receptor. And so this is Kd. Now, Kd, or the affinity of the ligand to receptor, is expressed as a proportion of how well the reverse reaction is over the forward reaction, where K2 is the reverse reaction, so the reaction goes this way, and then K1 is the forward reaction, where the reaction goes this way. And then the third equation describes that the number of occupied receptors is proportional to the max effect, and this determines the observed effect. And so considering a situation where we are comparing the ligand concentration for the observed max effect and the percentage of occupied receptors, this is what you'll see. So if this is a ligand concentration describing ligand concentration for percent of max effect, where you'll see max effect at this concentration. And then, however, to get all of your occupied receptors to be occupied, you'll require a larger ligand concentration. And so you see a max effect before a maximum is reached on receptor binding. And in terms of receptor binding for max effect, this concentration over here is also known as the EC50, where the EC50 is the ligand concentration, where you see 50% of the max effect. And corresponding to that same 50% of occupied values on this corresponding line, this is Kd, or the ligand affinity. So I'm going to talk about the regulation of receptors in the terms of desensitization and refractory periods, where the second dose does not always have the same effect as the first dose. And so if we administer a ligand or receptor and you see the max effect, and you want to see what the effect is from the second administration, different receptors will have different refractory periods. And so with a receptor that has an absolute refractory period, there is no effect on the second administration. And so this is in red that I put here. And now with the relative refractory period, there is a reduced effect, and maybe at some time later it might get back to the same response. So maybe there is a third administration. It might go back to where this max effect was, but on that second administration, or within that relative refractory period, the effect is going to be diminished. And then there's going to be hypersensitivity and hyperreactivity, where the subsequent dose is greater than initial effect. And you'll actually see this a lot in IgG levels after like the second vaccine dose, when you have a booster of vaccines. And so these are different ways that receptors can affect the max effect after the first administration. So I'd like to talk a bit more about ligands, but before that I need to explain to you the 4 different states of receptors. So receptors can exist in 4 different states based on their binding as well as their activity. And so when you have a receptor that's unbound, so it's just R, but it's inactive, that's a typical state that we usually find for ligand-bound receptors, that when they're not bound, they are inactive. And then on the flip side, when you have ligand-bound receptors, they are active. This is another very common state that we usually consider. However, there's also a case where we could have unbound receptors that are actually active. And so we actually define this as the basal activity, where when being unbound, they have the capability of acting in a defined active state. And this depends on intrinsic or basal activity. And so these are receptors that are not bound but can still perform actions of the active state. And then there's also a case where you have a ligand-bound receptor, but it is inactive. And so even though they're bound to their ligand, they are still not performing the behaviors of an active receptor. And so when I talk about these ligands here that are bound to receptors, there are 5 different behaviors that they can take, where you have a full agonist, where after it binds, it is always in its active state. So when you have a bound ligand to a receptor, it is always in this state of being active. A partial agonist is when it's more likely to exist in the active state, but some of them are still going to be in the inactive state after binding to a receptor. Now, competitive antagonists does not alter the activity of the receptor. They will inhibit, but they do not alter the activity of the receptor itself. Partial negative agonists, upon binding, they're more likely to be inactive rather than active. And then full negative agonists occur when upon binding they are always inactive. And so if there's no basal activity, then these last 3 cannot be distinguished. So to predict the binding activity of ligands and receptors, you need to know 3 things. You need to know the basal activity, you need to know what ligands are present, as well as the extent of the ligand to receptor binding that you have. And so these are 2 equations to help with that, where RA is the basal activity, is defined by the portion of the unbound fraction multiplied by the basal activity. And then the portion of inactive receptors are the unbound fraction minus the basal activity, or the receptors that, when unbound, are active. And so we have an example of a full agonist whose receptor's basal activity is 20%. It's a full agonist, and then the ligand to receptor binding is 50%. What will happen is that your unbound fraction is going to be 0.5. Oh, because the ligand binding is 50%. This also means that your bound fraction is also going to be 0.5. And so what this means is that because we know that it's a full agonist and we know that half of them are bound, this is going to be in the LRA space, where they're bound, but also they're all completely active. So this is going to be half of the proportion of these receptors are going to be bound and active. After that, because we know what the unbound fraction is, as well as the basal activity, we know that basal activity is 0.2 from here. And so we can find RA, which is right there. And so this is going to be 0.1. And so the portion of unbound receptors that are active is going to be 0.1. Now we know that because of LR binding, the portion of bound receptors is 0.5, and the portion of unbound receptors is 0.5. And so the number of bound receptors that are inactive is 0, and the number of unbound receptors that are inactive is 0.4. And so this is how we determine for ligand and receptor binding what these proportions are. And so we have another example where we have a basal activity of 0, we have a partial agonist, as well as LR binding of 50. We know that half of them are bound, 0.5, and then half of them are not bound. However, we also know that basal activity is 0. And so if they're not bound, we know they're not going to be active. And so this has to be 0. That means that of the inactive, unbound receptors, this has to complete this proportion. So this is 0.5. So we know unbound fraction is 0.5. This is 0, which equals 0. Now we can find Ri. Oh wait, no, we already found Ri. And so how do we find these bound proportions? Well, we know that it is a partial agonist. So it means that most of the receptors, when bound, is going to be positive or active. And so, oops. So what this means is that the active proportion has to be larger than the inactive proportion. And so I have this greater than sign. And so because we haven't really defined what this partial agonist is, this has to be a larger number than the inactive portion. And so for now, we can create an arbitrary number. And so we can make this 0.3, and we can make this 0.2 to finish our square. And so in this third example, we're going to make this a bit more difficult. And so if we have a basal activity, where if we have an unbound receptor, 20% of them are going to be active still. However, this time, we're going to put 2 ligands into the mix, where we have a partial agonist and a negative full agonist. And so for one of these ligands, when it's bound, most of the receptors that are bound to this first agonist are going to be active. And then when the receptor is bound to the second agonist, all of them are going to be inactive. And then the ligand receptor binding for each of these is 50%. So if each of them are each of them are 25%, that means that the total percentage of bound receptors is 50, or 0.5, sorry. And then the portion of unbound receptors is also 0.5. And so we can find Ra. We're going to do that again first, where we know that the unbound fraction is going to be 0.5. 0.5, and then we know the basal activity is 0.2. This means that Ra is going to be 0.1. So 10% of all of these receptors are unbound, but also active. Now we know that this inactive state of unbound receptors also has to match up with this proportion of unbound receptors, so we also know that this is 0.4. So that was the easy part. The part that makes it a bit more difficult is when we separate the partial agonists and the negative full agonists when we're talking about the bound receptors. So let's first talk about the negative full agonists, because that is a bit more easy to talk about. And so if you take this box down to over here, we know that if it is bound, they are definitely going to be inactive. And so for that 25% of receptors, we're going to have 0.25 and 0. And then if we look at the partial agonist, we're going to use the same partial agonist before, where it was 0.2 and then 0.3. So 2/3 of them, 2/5 of them will be will have an inactive state, and then 3/5 of them are going to have an active state. And so in this case, when there's only 0.25 of them, 0.25 of the total population that are bound with the partial agonist, this means that 10% of the entire population are going to be bound with this partial agonist and going to be inactive. And then 0.15% of the total population of receptors are bound to the partial agonist and are active. And so when you add these two together, 0.25 plus 0.1, and then 0 plus 0.15, you get the boxes here, where this ends up being 0.35, and then this ends up being 0.15. And so next time I'd like to start talking about the barriers to absorption that we find in drug delivery on a very human physiological scale. And thank you for watching this lecture. Thanks.